The materials that will be used for this DNA extraction will be a beaker, test tube with tight lid, pipettes, paper cups, salt, isopropyl alcohol with 70% concentration, and liquid dish soap. We will begin by creating the saline solution by adding 25 milliliters of distilled water onto a beaker. We will then add 2 lap scoops of salt and stir completely the water until the salt is dissolved. We will also add 25 milliliters of fruit punch Gatorade onto another beaker. We will then swish the substances in our mouth for 30 seconds. While swishing, we will carefully scrape our cheeks with our teeth to remove our cheek cells. After the 30 seconds are up, the substance goes back into the beakers and then transferred onto the test tubes. Make sure not to fill the test tube all the way up with the solution. You will now add 2 drops of the liquid dish soap onto just the saline solution, and also 20 drops of the isopropyl alcohol for both solutions, but you will fill up the saline solution with only 20 drops of the alcohol, and you will fill up completely the Gatorade solution with the alcohol. After adding the alcohol to the solutions, you will tilt them back and forth for a few times. Then you will seal them tightly and set them upright for about 1 minute so the solutions can combine. After the minute is over, you will be able to notice the DNA red clump in the Gatorade solution. In the saline solution, there will be more clumps, but they will be clear, and that is your DNA. The variables changed during this experiment where the Gatorade was turned into the saltwater solution, meaning that the salt in the solution allowed the cells to clump together. The cold isopropyl alcohol was easier to preserve the DNA. The soap allowed the DNA to be exposed by breaking down the DNA. In conclusion, our hypothesis was correct. Using clear liquids and the salt water did allow the DNA to be seen more clearly and bigger.